Okay, so I would like to thank everybody, the organizers, for the invitation. Yeah, it's really a pleasure to present for you. Of course, I would like to be there instead of here in front of my computer, maybe next time. So I'm going to speak about endometrial NK and plasma cells in infertile women. If it works, yes. NK cells are very important players from the beginning. You see here a blastocyst, which is reaching the endometrium. We are more or less in this area at day 4, 5, here day 6. And here we have the attachment and the implantation beginning with the invasion of trophoblast cells into the endometrium. And here in the endometrium, we find natural killer cells. They sound dangerous, but they're not really killers here. They are preparing the endometrium for successful implantation. But it seems that a certain number of NK cells is an optimum. So not enough or too many may be harmful for implantation and for the beginning of pregnancy. So you see here, many immune cell types are present in the endometrium. You see almost the entire immune system is present. You see uterine NK cells, T cells, B cells, but the strongest group is the uterine NK cell. You have also macrophages, and some of the NK cells seem to proliferate in the endometrium. Some seem to enter through the endothelium of the vessels. So I will speak about very briefly about basic NK cell functions. Then I will show you NK cell analysis in the endometrium, a study that we are doing, and plasma cell analysis in regard to chronic endometritis, also in endometrial biopsies. So peripheral NK cells, they are mainly cytotoxic. They have strong cytotoxic potential, and they are mainly characterized by the expression of the CD16 Fc gamma receptor. And this receptor interacts with immunoglobulin G, and this interaction can activate the cytotoxicity. It has a low expression of CD56, which is an adhesion molecule. It expresses killer cell immunoglobulin-like receptors, the so-called KIRs, which are important players in the interaction with HLA molecule and in recognizing self and non-self molecules and cells. And these cells have strong effector functions. That means they are highly cytotoxic and can be harmful for foreign cells or for cells which express different HLA molecules. And some of them express CD57, which are potent cytotoxic functions, especially in highly differentiated NK cells. So peripheral NK cells are strongly cytotoxic. In contrast, uterine NK cells, they also do express CD16, but at lower levels, very low levels, let's say. They express CD56 in high levels. They have a different KIR profile, but they express a high number of cytokines and growth factors. And they have effector functions, but they are not as strong as in peripheral NK cells. So they are strongly reduced, but they can be activated. In certain circumstances, under stimulation, they can be strong again and can be similar to those of peripheral NK cells. But normally, without any stimulus, they are much less active. 300 different genes have been described between these two cell types, the uterine and the peripheral NK cells, out of 10,000. And summarized, their function is immunoregulation, angiogenesis, so the production of the different cytokines and growth factors, so especially growth factors like VEGF, which are related to angiogenesis, and they have a low cytotoxicity. So this is again the endometrium, but now a decidua has been formed. This is in early pregnancy. Here you see trophoblast cells invading from the anchoring villus. So here on this part begins the fetus, the embryo. These trophoblast cells invade here, the decidua, and are in direct contact with the different immune cells. And this contact is not dangerous and not harmful for the trophoblast cells. It's a kind of interaction and communication between the different cell types, between trophoblast cells of fetal origin and immune cells of maternal origin. In some cases, trophoblast cells can die, fragment, or can undergo apoptosis, necrosis. Fragments can be taken up by macrophages, can be presented to other immune cells, could activate immune reactions, can also activate regulatory immune reactions, Treg cells. So it's a very complex system. And to my impression, the equilibrium, the right equilibrium of the numbers and proportions of cells in the decidua is fundamental for successful implantation and then for successful growth of the placenta and successful pregnancy. So very briefly, 2 slides with some experiments that we are doing, just to show you that we are not only measuring, this is the main part later on, that we are also doing experimental stuff in our lab. So for example, we are analyzing the interaction between trophoblast cells and immune cells, in this case, natural killer cells. And for this approach, we use trophoblastic cell lines here, and we transfect these cell lines with microRNAs, which are not originally expressed in the cell lines, but in the original trophoblast cell. So there are some differences between cell lines and primary trophoblast cells. And we have cell lines which lack these fundamental microRNAs, fundamental for trophoblast cells, and we transfect them into these cell lines. Then we culture these cell lines. We isolate microvesicles, that means small or large, in this case, large extracellular vesicles. And then we incubate recipient cells, for example, NK cells, but also other immune cells in the completely different cell types from different organs with these microvesicles. And then also with exosomes, with the small extracellular vesicles. And then we detect these microRNAs in the target cell, in the recipient cell. We analyze the function of these microRNAs in the target cells, and to mimic here the communication between between fetal cells and maternal cells. And this is what we guess what is happening in the decidua, in the placenta, and basically in the entire body of the woman, of the pregnant woman. And as you can see here, in green, these are green stained microvesicles. Blue, this is the nucleus of NK cells, and these NK cells have taken up the green stained vesicles. And here, this is one huge nucleus. This is with 2-photon microscopy, and you see here vesicles around the nucleus. This is an NK cell, and vesicles derive from these incorporated vesicles derived from trophoblast cells. And just to show you one slide, what they can deliver here, this case, so again, trophoblast cells or trophoblastic cell lines have been transfected with microRNA 519. We've isolated different types of extracellular vesicles, and then we have incubated immune cells with these vesicles. Then we have done a qPCR to detect this microRNA. And as you can see here, when we transfect immune cells with this microRNA, we have like 10 million-fold increase of it. And then when we incubate immune cells with extracellular vesicles from trophoblast cells, we have also a very strong, significant increase of the microRNA, which we have transfected here in this origin cell. So up to, in this case, up to 10,000-fold when we use the large extracellular vesicles. This is just to show you rather novel example of communication between trophoblast cells, and maternal immune cells. So I told you we are analyzing NK cells in the endometrium in patients with infertility problems. We have been motivated because of this paper here from 2013, which is a meta-analysis which does not really show clear results. It indicates elevated NK cells in case of a recurrent miscarriage, but we saw here very low numbers of cases. N is always below 100, and the results were not very stringent, but indicated increased NK cells in miscarriage. So we have collected endometrial biopsies. So this is by the end of 2020, by the end of last year. Meanwhile, we have 4,000 samples more. So we are at almost 18,000 samples. In 11,332, we have analyzed uterine NK cells. In 8,742, we have analyzed also plasma cells. And we have received these samples from more than 200 IVF centers, mainly in Germany, but also in other European countries, mainly Netherlands and Norway and Austria. We have, in the beginning, we have defined normal values. This has been established on fertile patients. So the first group were patients who underwent some gynecological tests in the University Hospital of Heidelberg. And then we confirmed these values on a group of 27 oocyte donors from Spain and from Ukraine. And we stayed after this with these values. I show you the distribution later in another slide. So this is how it looks like. Endometrium with a very low number of NK cells. Here the blue nuclei are stroma cells. And you see here an example of strongly increased uterine NK cells. The brown staining shows the NK cells. And you see just by eye, without counting, a huge difference between the different groups here. And our estimation is that between 40 and 300 uterine NK cells per square millimeter of the sample is the normal value. So this is the distribution. This is the number of samples in a certain interval of cases. So this shows here from 40 to 300 NK cells, and this is in steps of 10. And you see most samples are here. Then it's going down, and we have patients with up to 1,500 NK cells per square millimeter. This is the distribution currently. Unfortunately, I cannot see my own image completely. So here in the patients, we had approximately 5% of samples with low numbers of NK cells below 40. In the controls, 1. Then we had 7,300, which is approximately 65 in this normal, the 65% in the normal range. Then approximately 30% in the elevated range, and 9% in the highly elevated range. And you see the distribution of the normal controls. So I told you we have defined it, and then we did not move it. And we saw that also in these healthy patients, in these healthy donors, we had also one case with low numbers and 2 cases with elevated numbers. This was a side analysis which we have done on a group of donors from Ukraine, and we saw a difference in NK cells, in endometrial NK cells, in a natural cycle and in a stimulated cycle. So it's not significantly different because the number of cases was small, but it indicates elevation of NK cells under stimulation for preparation of IVF. These are results of a study that we have done together with partners in Heidelberg University. We have analyzed the numbers of uterine NK cells in patients with more than 3 idiopathic recurrent miscarriages, and we have excluded a lot of different factors. You see anatomic disorders, thyroid dysfunctions, autoantibodies, thrombophilia, chromosomal disorders. And at the end, we had 58 patients with idiopathic recurrent miscarriage. And the results were the following. The mean number of uterine NK cells, approximately 250. And this is the standard deviation. And this is very typical in all our samples. We have huge standard deviations and a very broad variance. But you can see here the difference between the patients and the controls. And this was statistically significant. Then we have used our categories. And you see here in our controls, we have 5.9% of samples with low values, 88 with normal, and also 5.9 with increased numbers of NK cells. But in the group of patients with recurrent miscarriages, we had only 62% with normal numbers and 34.5 with increased NK cells. And this is the current status of all patients that we have. So they are not filtered for recurrent miscarriage. This is simply all we receive from the different centers with different reasons for infertility. And we see here also a lower percentage, lower proportion of elevated NK cells than in this very exactly defined group of patients. Here I would like to show you the dependence of the day of cycle, so the number of uterine NK cells in the endometrium per day of cycle, or depending on day of cycle. And the blue bars show the mean and the orange bars show the median. And this is the regression curve here. And you see, we were really surprised when we saw these results. I told you about the broad variance of results. But anyway, there was a very clear increase of the mean of NK cells day by day during the menstrual cycle, the ovulation starting from day 14. And this is based on a huge number of samples. So you see here under the curve, you see the numbers that we have analyzed per day of the cycle. So for example, at day 21, we had 1,504 samples. So these results are extremely robust. Okay, I move to plasma cells and chronic endometritis. If you check literature, especially the older literature, the incidence of chronic endometritis in recurrent implantation failure and recurrent miscarriage was extremely variable. So nobody really had a good definition. So between 7 and 67% of cases of recurrent miscarriage was described as having chronic endometritis. So the problem was the studies were small, the definitions were different, and the methods were also different. Usually the diagnosis is done has been done by hysteroscopy. Nowadays, the standard is to analyze CD138 positive plasma cells. And this has been used in several previous papers. But the problem is that these papers use for example, different fields. Some define the surface of a sample, so like we are doing it now in square millimeters. Others just count cells in high power fields. I try to find out the variation of the size of high power fields. So it is really, it's dependent on the objective of the microscope, and it can vary by a factor of 20. So it's very hard to compare. If you read one paper on high power fields and another one on high power fields, it is not the same. Then some groups have measured also NK cells in blood, or they have dissolved or disrupted the endometrium, the structure, and then they have produced a suspension of cells and analyzed the suspension by flow cytometry. So there are many different methods, and it's very hard to compare. So it has been supposed for a long while that the endometrium is sterile, but in more or less recent studies, it has been shown that there are also bacteria, at least RNA, bacterial RNA, as shown by qPCR or by next generation sequencing. It seems to be much more difficult to do bacterial cultures from endometrium, and there are also some more recent papers and reports I've heard at conferences which say it's absolutely impossible to culture bacteria from endometrium. So I think this discussion is still open. Some authors have tried to define specific bacteria responsible for chronic endometritis, and they have tried to treat very specifically with specific antibiotics, but this was not as successful as using broad spectrum antibiotics. And they also found that the presence of these specific bacteria is not sufficient for having a chronic endometritis. So the reason is unclear. Some patients have it and others don't have. This is the Japanese study. Here they have analyzed a lot of different bacteria in patients with and without chronic endometritis, and they found one single significant difference in Corynebacterium, which was elevated in patients with chronic endometritis, but this was only in 8 out of 105 analyzed cases, and this without chronic endometritis in 1 out of 251. So it's a very rare event. It's not sufficient to explain the chronic endometritis in general. And in the others, there's no big difference, nothing significant. So currently there are controversial studies by several groups concerning the influence of the presence of different lactobacilli, Einers, Crispatus, or Gasseri, and others. So their composition seems to influence the flora and the susceptibility for a chronic endometritis. So I have shown you this before, and again, here are the different immune cells in the endometrium, and in this I would like to add the plasma cells. Plasma cells develop from B cells, especially from memory B cells, so by passing through several steps, and they are searching or finding survival niches in the body, and these are always sites of chronic inflammation. So this can be the nose or the skin, or it can be the endometrium. And usually the presence of the plasma cells indicates a chronic inflammation. They should be absent in healthy tissue. So the definition of chronic endometritis has been very different. You see here in some papers, at least the presence of at least one single plasma cell has been defined as chronic endometritis. Other groups have said more than 4 plasma cells per level, but not clearly defined, but only 0 plasma cells are normal. So it's heterogeneous what we found. And we have decided to do the most conservative estimation. So we said over 4, so starting from 5 plasma cells per square millimeter, we have a chronic endometritis. But there's a recent paper, which I like very much. It's from a Chinese group in Shenzhen, and they have analyzed in samples from patients with chronic, from infertility patients, 30 high power fields under the microscope. Unfortunately, it's not defined how big they were, but so this is how it looks like. And then they found in the different high power fields different numbers of plasma cells. And they have counted how many cells there were per field. And they have been focused on the field with the highest number of plasma cells. So for example, in this case, it's this one. This has 5. So here we have a patient with at least one high power field with 5 plasma cells. And this was there's another one, and this was their definition. So if they found more than 5 plasma cells in one high power field, they had a reduced live birth rate. In all the others, when they found only nothing, so no plasma cells, or maximum of 1, 2, 3, or 4 in one of these 30 fields, then they had a higher live birth rate, between 58 and even 83%. But if they found more than 5 in one high power field, then the live birth rate was reduced. And this demonstrates a little bit or it reflects the inhomogeneous distribution of plasma cells in the endometrium. So probably it's not a reaction which is the same over all the endometrium. It seems to be locally increased. So there seems to be areas with more inflammation and with less inflammation. And if there are a few regions with a strong inflammation, this seems to be disturbing implantation and live birth rate. So I recommend this paper from Lee et al. From last year. So I show you a few of our own data. So jump over this one. So we have analyzed the numbers of plasma cells, and you see here again intervals of numbers. So this is 0, then 0 to 2, 2 to 3 to 4, 5 to 6, 7 to 8, and so on. And of course, most of the samples that we have analyzed are in the range between 0 and 4 plasma cells. So this is normal. And then we have also patients with elevated plasma cells. And I increase this part with a highly elevated. And you see we had here more than 100 patients in this group with 11 or 12 plasma cells. And so on. So this is the number of cases in each of these categories. And what you can see here is we had also patients with more than 100 plasma cells per square millimeter. And this was highly interesting. And some of them had it even after antibiotic treatment and did not change. And I remember 3 patients that we had, and we have contacted the doctors, the clinicians, and one of these patients had HIV, one had leukemia, and one had immunoglobulin M syndrome. So all of them had some very strong disorders or diseases behind that. So the endometrium seems to be able to reflect general diseases. So what you can see here, this is the number of normal samples, 77%. This is elevated and strongly elevated numbers of plasma cells. So we have approximately 23% of our samples have elevated plasma cells. Here you see the dependency on the day of cycle. And here you see that the mean number of plasma cells that we found per square millimeter is higher in the proliferative phase before ovulation than after ovulation. But there's not such a clear day-by-day dependence as for the NK cells. And this is just the distribution of all values. Then we have compared the number of plasma cells before a treatment and after a treatment. So this is the pre-treatment number. So starting from 4 plasma cells, this is what we, starting from 5, this is what we say, this is a sign for chronic endometritis, and then up to 80 plasma cells here. And these patients have received doxycycline therapy. And then we have checked for the change of plasma cells per square millimeter. And you see here, this is a reduction of plasma cells. So 25 plasma cells minus 50 plasma cells less, or here increase of 25. And you see a clear reduction of plasma cells after antibiotic therapy. So this is shown here. So 414 patients had a decrease, 12 patients had no change, and 71 patients had an increase of plasma cells after the therapy. We have also checked what happens to the NK cells after the antibiotic therapy in only 20 patients, a very small subgroup here, and they have been decreasing also. So these are numbers that I've shown you before. These are 5,800 patients which we have analyzed. 77% were normal, and these are the elevated and strongly elevated values. Oops. This is what we recommend for treatment, 200 milligrams daily during 2 weeks, so twice 100 milligrams per day. If this is not efficient and we see again elevated plasma cells after this treatment, we recommend metronidazole 500 milligrams, and previously we have recommended ciprofloxacin, but because of potential side effects, we are now recommending amoxicillin and clavulanic acid. These are very recent results, therefore the numbers are higher than in the previous slide that I've shown you. This is very new. So again, comparison, plasma cells analyzed, meanwhile 8,700, and you see here not reduced and reduced numbers of plasma cells after therapy. So out of these 2016, which had high numbers, these are the numbers which have not been reduced after the therapies, and these are the numbers that have been reduced after the therapy. So that means we can show here that antibiotic therapies really reduce number of plasma cells. Almost at the end. This is a study which has shown increasing ART live birth rate following antibiotic treatment. So when patients have been successfully treated and had no chronic endometritis after the treatment, the live birth rate was far higher than in persistent chronic endometritis. And even the natural live birth rate following antibiotic treatment has been increased very strongly. The same group, Ciccinelli, compared to patients with unsuccessful treatment or with unknown infertility reasons. So antibiotic treatment seems to improve the live birth rate. Summary: endometrial natural killer cells are controlled by trophoblast factors. Increased concentrations may disturb implantation and placentation. Therapy options, I did not speak about them. I thought there might be some clinicians here in this meeting who will speak about this: corticoids, intralipids, and intravenous immunoglobulins. And plasma cells indicate chronic endometritis and can be treated with antibiotics. And yeah, thanks for cooperation, especially, I don't read every name, and especially also for our group in Jena. And I would like to thank you for listening. Thank you.
Thank you very much, Professor Markert, for this wonderful presentation, and congratulations for the huge number of samples and patients you have collected, especially endometrial samples, because this is a huge biobank for the future investigations on this really extremely important topic. Now I give the opportunity for the audience to put questions on this topic and on your presentation. Do we have questions from the audience?
Katya, yeah, we have Professor Kirchev.
Sorry. Can you hear me?
Yes, nice to hear you.
Udo, it's nice to meet you again. Unfortunately, we cannot meet personally. I have seen that you have analyzed NK cells by immunoperoxidase reaction. Have you tried any other method? Let's say flow cytometry. I'm asking this because recently Jan Brossen's group published a paper saying that they have analyzed, measured in any way, the NK cells, endometrial NK cells, by flow cytometry. Knowing that, well, for flow cytometry, you need a lot of cells, a lot of numbers. I was wondering whether anyone has ever tried to use flow cytometry to detect and measure the uterine NK cells. Have you done this? Have you tried this?
So we are doing only immunohistochemistry, and the reason is that we receive samples from all over Germany and Central Europe, and they just take the samples, put them in formalin, and send them by common mail, by just normal mail, even from abroad. And so that takes sometimes one day, sometimes a few days, but this is a very safe way, and we receive always the same quality of samples. And for this would be much more difficult for flow cytometry because, yeah, you need to produce suspensions. This is something that you could do or that you can do in your place with your own samples, maybe, you know? But for sending, this is really difficult. But we have a partner in Ukraine, and probably you know the people, Ksenia, with a very complicated family name,
yes.
and Boris Donskoy. And we have compared, they have analyzed their samples with flow cytometry and we with immunohistochemistry, and we had the same type of results. So they have seen, for example, elevated NK cells in stimulated cycles compared to natural cycles. And the quality of results was the same that we found. Yeah. I mean, it has the advantage, flow cytometry, that you can calculate portions of cells. You can really say that 2% or 3% are NK cells or something like this. On the other hand, to my impression, it may be also, or it seems to be also important how big your sample is, or if there is also space in your sample without cells, cell-free, and which may be edema, or which may be mucus or huge glands or something like this. And I think it's the density of cells over the volume, which may be interesting. But I agree that flow cytometry is of course also interesting.
And still another question I have you noticed some specific location of the plasma cells, let's say around vessels or something like that?
We see especially So usually we get endometrium, but in some cases, accidentally, we have cervix epithelium. And then the epithelium itself is positive for CD138, which is also kind of a positive control. But especially in the cervix, near the epithelium, there we see higher numbers of plasma cells sometimes. But inside the endometrium, to my impression, distribution is really random. It's here and there. I've never seen a clear, clearly preferred localization.
Thank you. It would be nice to meet you again. Please come next summer in Borovets.
Yes,
we have a national congress, so I will invite you again. Is it okay?
I try my best. I hope. Yes, I accept this pleasure, of course.
Thank you. Thank you.
Thank you.
Thank you very much, Professor Markert, for the wonderful presentation and wonderful work, and see you next year in Bulgaria. Thank you.
Thank you.